so I cannot stay behind this column, so I don't know where to stay. Yeah. Anyhow, thank you very much for this presentation. Thank you for coming here instead of going to the beach. I said yesterday the beach is very crowded. So I want to entertain you today about a fascinating feminizing bacteria. I don't know if it's the place to speak about it, but let's say this is this convention. It's fascinating because it opens new therapeutic and preventive avenues for filarial and some arthropod-borne infections. And what you see down is the future University Hospital Center in Montreal is not yet achieved, and the Wolbachia is there. My objectives for today are just three. To have basic information on this pandemic under symbiont, to learn more on filarial nematodes infection with Wolbachia and the new therapeutic avenues, and to understand the arthropods infection with Wolbachia and the new preventive approaches for the control of some vector-borne disease. I have no conflict of interest of any kind. In order to address the three objectives, for the first one I'll speak a little bit about the biology and the virulence factors of Wolbachia. For the second, I will give a few data on the existence of Wolbachia in filaria, a few words about the pathogenesis and the effect of antibiotics, and then I'll speak a little bit about the treatment of filariasis, devastating disease. For the third objective, I'll speak a little bit, and it's a little bit complicated, but do not panic, about the reproductive strategy of Wolbachia in insects, and then I'll speak about the vector and disease control. There is an increasing interest, especially in the last 5 years, on Wolbachia because it's widespread and it's extremely abundant, you'll see. It has fascinating effects on the reproductive biology of its host to assure its dissemination and survival. And mostly because there are practical current and future applications in the treatment and prevention of several filarial diseases and also in the prevention of some vector-borne disease. You may see that it's an hereditary pandemic because it goes from mother to eggs. I'll explain to you. And it's pandemic because 65% of the insects are infected, which means 2 to 10, depend on the author, million infected species of insects which are infected. And also other arthropods like Crustacea and Cerates. And then you see here on the right the nematode, especially from the filaria group. It was firstly described like rickettsia-like organism in Culex, the mosquito, Culex pipiens, in 1924 by Hertig and Wolbach. Then it was classified in the kingdom of bacteria, and it's an alpha proteobacteria, a very ancient bacteria, millions of years, related to rickettsia. In 1936, Hertig classified in the genus Wolbachia, the unique genus, with one species which was Wolbachia pipientis from Culex pipiens. He gave the name Wolbachia to honor his mentor, who is Dr. Wolbach. By the way, he spent a year of his residency training in Montreal, but not under my direction, believe me. For the phylogeny, you may see here that it's related to Ehrlichia, Anaplasma, and Neurickettsia. There are a lot of families, but A and B are very well described in arthropod since 1924-26. And then those who are in green are described in filarial nematodes, but 50 years later. The type of symbiosis. With filaria, Wolbachia has a mutualism, so it's beneficial for both Wolbachia and filaria. In arthropods, could be either commensal or even parasitic. And we'll move on. It's a gram-negative intracellular bacteria. This is called, it's a symbiont, and it's called endosymbiont because it's intracellular. It will be probably in a million years from now on another organelle like mitochondria, which originally were the gram-negative bacteria. It's a vertical transmission, as I said, from mother to offspring, but rarely also horizontally from insect to filarial nematodes. Infects in general the cytoplasm which surrounding the germline stem cell in insects. And this will give rise to several reproductive phenotypes. I will explain to you, so don't panic. But also it could infect the somatic tissue, both in filaria and insects. It has a small genome, and the complete sequence is known for Wolbachia from Drosophila melanogaster, which is the fruit fly, a very small, how should I say, fly which is very well studied. And also for Wolbachia from Brugia malei, which is a filarial nematode. It has some biosynthetic pathway, and there are others which are absent. So it will take what it needs from the host and will give to the host what the host doesn't have. So the glycolysis is present. The pathways for the synthesis of purine, pyrimidine, riboflavin, and HEM are also present. What is more important is that it has some major surface protein A and B, which are antigenic, immunogenic, and also pathogenic factors. And we'll see later on their implication in the disease. It has also proteases, other protein like heat shock protein 60, catalase, which will protect the host, the filarial worm, peptidoglycan-associated lipopeptide, endotoxin, and especially the neutrophil chemotactic factor. I had here a link, but doesn't work here, to give you the whole, the access to the whole data on the genome of Wolbachia. And I tried tomorrow and it doesn't work, it stops my presentation. I don't know. So I finished with the first objective, and I summarized the basic information here. It is an ancient bacteria that became an intracellular symbiont and is widespread in invertebrates. Cannot exist outside of the host, utilizes the host reproductive system for its reproduction, survival, and dissemination. There is a mutualist symbiosis in filaria and mostly parasitic symbiosis in arthropods. Now I'll move to the second objective, which is Wolbachia and filaria. It was firstly described in filaria in 1975, so 50 years after it was described in insects. It was described like intracellular bacteria at electron microscopy. But these authors had the foresight to say that these intracellular bacteria resemble to those which were described in insects. In addition, and I was impressed, they said probably this could be treated with antibiotics. But not many people learned their article, I think, because only 20 years later, similar organisms have been described in Dirofilaria imitis, which is the hard filarial form in dogs, and also in cats. So 20 years later. But what is ironic, it was that Wolbachia, it was used for the prevention of filariasis, or to diminish the incidence of filariasis. And it was a proof of concept published in 1967, showing the eradication of the local mosquitoes, Culex pipiens, by replacing them with another one which is naturally infected with Wolbachia. This proof of concept stayed there for a long time. No one tried to repeat it. Except 44 years later, it was used to do the same in the Southwest Indian Ocean Islands, and then also in the South Pacific Islands. But this was very nice experiment. Aedes polynesensis was the local vector for lymphatic filariasis. They caught the mosquitoes, they treated them with doxycycline, they reinfected them with different species of Wolbachia, and they released them in nature, and they replaced the other population. Okay? So it was an extremely nice experiment, and it was repeated twice. Almost all filarial species are infected, as demonstrated by electron microscopy, immunohistochemistry, or PCR. However, there are some of them. Here are the filaria species in humans, and this is in animals. You may see Loa loa, which is not infected. However, when you look at its genome, you'll see fragments of gene very similar to those of Wolbachia. And the people are saying that probably Loa loa was infected before other filarial hosts, and it was able to get rid of Wolbachia. The same also in animals, in cattle. You may see Onchocerca flexuosa, which is not infected. Now the most important disease caused by the filarial hosts are onchocercasis, or liver blindness. You may see the environment, which is very nice, where the black fly, which is a vector or similar, is a vector of this parasitic infection. You may see the filarial worm, the adult worm, the life cycle, and also the clinical manifestation. First of all, a lot of nodules or onchocercoma, which I don't know why. They are more frequent in the upper part of the body in America and in the lower part of the body in Africa. There are also other cutaneous manifestations, but most important is the liver blindness, is keratitis, and chorioretinitis. What you see here is the statue erected in front of the World Health Organization in Geneva in 1999 to celebrate 25 years of drug mass administration of ivermectin in their fight against onchocercosis. And it represents what you see in the villages where the onchocercosis is. Adults will leave the village and will let there only the infants and their grandparents, which are already blinded, to go to work in the field, ready to do. So another statue will be erected probably in a few years from now on. What happens with Wolbachia and Onchocerca volvulus? It infects all individual parasites at all their development stages. It's endosymbiotic mutualism, I told you, and it's both nutritional. Wolbachia furnishes the nutrients for Onchocerca and vice versa, but also defensive. You've seen that Wolbachia have several proteins, catalase, so this will protect the worm against the human defense. So it compensates for the absent or incomplete biosynthetic pathways in Onchocerca volvulus. It has also immune and pathogenic effects on the human host. And we thought many times that what we see in the humans is due to the filarial worm. Well, this is not true. It's due to Wolbachia. Because the filarial worm, it's like in America, they are called CD. CD means cross-dressed. So this is a worm in the, how should I say, dressed, yes, Wolbachia. Because Wolbachia's surface proteins are on the surface of the filarial worm. And what is most important from a practical point of view, you may eliminate Onchocerca volvulus totally by treating Wolbachia with antibiotics. And I will show you later on. Now, the first manifestation I told you is the nodular onchocercoma. Here you may find both microfilaria, which is the larva, and the adult filaria. And to illustrate what I told you, there are in red Wolbachia surface protein. You may see it in both microfilaria, in both the reproductive system and in the somatic tissue. And also you may see adult filaria which is infected with Wolbachia. And to illustrate the pathogenic role of Wolbachia surface protein, you may see here that peripheral blood mononuclear cells from healthy volunteers in the presence of this protein will induce the secretion of inflammatory cytokine TNF-alpha, IL-1, IL-6, IL-8, and this is dose-dependent. In onchocercasis, when microfilaria dies, it will release the Wolbachia, and through the toll-like receptors and others will activate the fibroblast, dendritic cell, and keratinocyte, which are present in the cornea. They have also the neutrophil chemotactic factor, and they'll go there in the cornea. It will be an accumulation of neutrophils and also the macrophages, and this will give rise to corneal haze and cystic. But Wolbachia could also be liberated from microfilaria when you start the treatment. And the usual treatment was diethylcarbamazine and ivermectin. So the Wolbachia is released in the blood. And we know that when you start the treatment, you may have adverse effects, adverse reactions, either local, cutaneous, or systemic. So what will have the Wolbachia will increase the production of cytokines and antibacterial peptides. It will be accumulation of neutrophils, and this will give rise to the skin manifestation. I told you that in onchocercoma, in the nodule, you may find both microfilaria, the larva, and also the adult. You may see the adult, the section in the adult worm. What is in green, I don't know if it's green, but from my, in my PowerPoint is green. This is Wolbachia here. And this is the somatic tissue, and this is the reproductive tissue. So you may see the adult worm, which is infected with Wolbachia. But in the onchocercoma, you see again the adult worm, which is intact. The Wolbachia is still inside, but there are a lot of neutrophil infiltration. Illustrating what I said before, this worm has closes of Wolbachia. So it has the Wolbachia surface protein on its surface. What happens if you treat Wolbachia with doxycycline? So you treat it, you may see that the inflammation disappears, the nodule will resolve. But what you'll see again is that the adult one has no more Wolbachia, neither in the somatic tissue or reproductive tissue. There are other filarial infections like with Buchereria bancrofti, which is responsible for the lymphatic elephantiasis and the lymphedema. And also I told you in the dog and also in the cat you may have this heartworm, which is Dirofilaria immitis. And we see that even here there is a temporal association between the beginning of lymphedema and the development of immunoglobulins IgG or antibodies against the Wolbachia surface protein before the specific antibodies to Wolbachia will be there. And I had patients starting with lymphedema. Their test for filaria was at the limit, was not entirely positive. I didn't think at that time to measure this one. It was only research. This was done in Haiti. However, I treated the patient, everything disappeared, he's very well, and then I tested him for filarial, and he's positive, so it confirms the concept which is illustrated here. What are the effects of antibiotics on Wolbachia and filarial? There are studies in vitro showing that both all this antibiotic listed here, tetracycline, rifampicin, azithromycin, tigecycline, active against onchocerca and other filaria like Brugia, Buchereria, and Bilofilaria. They are macropilaricidal. They kill the adult worms. It's the only drug that kills the adult worms. With albendazole, ivermectin, and ethylcarbamazine, you don't kill it. You kill just the larvae. It blocks embryogenesis and the fertility and diminished the viability. This was in vitro. In cattle, it was also demonstrated that when you treat the infected cattle with Onchocerca okenji, with oxytetracycline, it's macropilaricidal, but eliminates also microfilaria. And the onchocercarial nodule, which are similar to those we find in humans, disappear 9 months after treatment result. There are, how do we explain the effect of antibiotics? You may see here microfilaria. What is in red is Wolbachia. This is the reproductive organs, and this is the somatic tissue. This is the non-treated worm or infection, and these are those treated. The difference is that after treatment, you don't see red spots here. All the Wolbachia disappear. But you see a lot of green spots, which is apoptotic nuclei. So the antibiotics in Wolbachia destroyed, but induces an excessive apoptosis, so the adult worm will die, and also the larva. There are some studies in humans. Firstly, the open studies with doxycycline for 6 weeks. And the people also said they will give a dose of ivermectin at the end, just in case there are some macrofilaria which are. And they seen with once a day, not twice a day, half of the dose. Wolbachia was eradicated in 90% of the cases, was macropilaricidal, you don't see any other adult forms. Embryogenesis was interrupted after 4 to 6 months, and no microfilaria. And there is now a consortium which is called Anti-Wolbachia. It's written here in red, Anti-Wolbachia Consortium. And they are trying to experiment with a lot of other antibiotics just to be able to shorten the duration of treatment. As I told you, when I was, before leaving Montreal, I had this link to the internet, but here I wasn't able in the morning, and this stops everything. Yeah. But there are not only open studies, there are also double-blind randomized studies with doxycycline plus or minus 1 dose of ivermectin, either 100 milligram per day for 5 weeks or 200 milligram per day for 4 to 6 weeks, macropilaricidal, sterilization of female worms. The people said, okay, 4 to 6 weeks is a little bit long. Let's give a double treatment with rifampicin and to give it to 2 weeks. But after 2 weeks, it was macropilaricidal at 60%, not 90%. But they didn't study it later on. But all the female worms that could be isolated, those 40% who still were there, were sterilized. And there are some other studies. I told you that ivermectin is used for more than 40 years with drug mass treatment. And there are people who receive it every 6 months to 12 months, maybe 17 courses, and they are still effective. So what they had, they tried, they used doxycycline 100 milligram per day for 6 weeks in those patients with persistent microfilaria after ivermectin. They seen the same. macrofilaricidal, female home sterilization, no microfilarial. And the people said it's a little bit difficult to do it in Africa. And there are a recent study, I think it was this year or last year, and they showed that this could be done, it's community-directed strategy. They demonstrated that it's feasible, and they looked even at 2 years what happened after the treatment of other people in the community with doxycycline, and the efficacy was present there at long term. So the proof of concept, not proof of concept, the proof is there. This is also true not only for onchocercasis but also for lymphatic filariasis. There are some studies using doxycycline 200 milligram per day plus 1 dose of the ethyl carbamazine this 4 months later to get rid of the persistent microfilaria. And also with rifampicin for 2 weeks, and this was very active. Eradication of Wolbachia, macrofilaricidal, clinical improvement. And here I have, this is a hero cell, and here are the filaria. They move very nicely. All the spikes here are their movements. I had a video clip showing their dance. It's called the filarial dance. I had also the music, drums, Hungarian dances. However, I do not have access to them. And what they showed after treatment, you don't see any coloration, Wolbachia, either in the epidermis or in embryo analysis. However, it was present before the treatment. So, the African Council for the Treatment of Filariasis, they decide we'll test and we'll treat all the person. Easy to say, and now it's easy to do, because there are some low-cost mobile phone microscopy with a reverse mobile phone camera. And the images they presented with this camera are very nice. How should I say? I didn't see in my microscope images like this. So this is feasible. Test and treat. So I will summarize what I said for the second objective in 4 lines. Wolbachia has a pathogenic role in onchocercasis and lymphatic filariasis. It is a proven target for the control of human and bovine cattle disease. Tetracycline is safe and effective in killing both adult and larval orbs. In spite of the quite long duration of treatment, is feasible and accepted by communities. And now, in insect, this is another story. And one of the researchers tried to think like Lady Gaga, but saying Wolbachia, Wolbachia, and he put in his song all the, how should I say, all the words I will tell you later on the reproductive. It was very nice, instructive. Once again, I have not access, but it's easy to find on YouTube, Wolbachia. You just write Wolbachia and you find it. It's very nicely illustrated. And these are the famous reproductive phenotypes. Don't worry. Here in, no, I didn't have it. Here is the filaria, and you don't speak about it. What happened in insects? It depends on the species of the insect. There is feminization. So an infected female, an uninfected male will give rise to infected females. And they have a feminization gene which is attenuated by the infection with Wolbachia, so they will be hermaphrodites. Some of the males have a feminization resistance gene, and they persist. They are very precious for the community here. So then the equilibrium will be started. In other insects, they do not need the males. There is a parthenogenesis. They divide it before the division of the cell. So it will be only infected females. What is a little bit sad is the male killing. So the infected female, when it's in contact with uninfected male, will give rise to the infected females. However, the egg here, because of Wolbachia, has like a toxin, and so the egg will not develop, and they will die. So the males will die. And even more sad is that those infected females, in order to develop more quickly, they will eat the infected dead males. Yeah. Other insects are a little bit nicer. The infected female, uninfected male will give rise to the infected female and also the infected males. Why? Because they are very, how should I say, attentive with the infected males. They have like an antitoxin to put there to protect against killing. And finally, the most important, this is called the cytoplasmic incompatibility, is here on the right. An infected female and an infected male, they are very, very much attractive. Yes, I wanted to point it out here. However, their progenitors will die because the uninfected female has no antitoxin to put there. So this is the concept that will be utilized. And it is here. Infection of arthropod vectors with Wolbachia effects with practical application. You may see here cytoplasmic incompatibility. Wolbachia carrying male and an infected male. I have not to look to you because if I look to you, I'll speak French. Sorry. So this is a wild type female. What will give rise, as I showed, is the embryonic test. This will reduce the population of these mosquitoes. In other reproductive phenotypes, the infected female and the uninfected male, the females will be resistant to different pathogens. So when they got some viruses like dengue virus, chikungunya virus, West Nile, and even Plasmodium, they are resistant. These viruses could not multiplicate very quickly in these insects. And then others, they have a reduced lifespan. So this will diminish their vectorial capacity. So the number of patients infected in a certain days. There are insects which are naturally infected, which is like commensals, like Culex, Aedes. Look here, it's also Glossina palparis, which is a vector for trypanosomiasis, the phlebotoms for Leishmania, and others. And others have been also infected experimentally. So like you see Aedes aegypti, which is not naturally infected, was infected with Wolbachias from Drosophila melanogaster. This diminished the life duration and give resistance to infection with dengue, chikungunya, Plasmodium, Brugia malaria. And this affects the feeding capacity, and I will illustrate it just after this slide. In Anopheles gambiae, it diminished the oocyst counts of Plasmodium falciparum and also Plasmodium verbi. Concerning the Aedes aegypti, you may see the quantity of blood they could have, because the proboscis here doesn't go directly. It has a lot of difficulties to get the blood, so it will diminish this vitality. And in Anopheles gambiae, infected with Wolbachia, and they use 2 strains of Wolbachia to infect it, you may see the oocyst count is drastically diminished after Anopheles was infected with Wolbachia. I don't know if I speak here a few minutes. And there are others which have been infected experimentally, and this gave rise to other effects. For example, Aedes albopictus, which is now the main vector for chikungunya and dengue fever. Dengue virus transmission is blocked. In Culex, there is a protection against West Nile virus infection. And there are a lot of studies in the field, and this is Scott O'Neill in Australia. He has here a lot of mosquitoes. They've been infected and released every week here, and then they dominated the population. And he has the approval to do it in 2 villages. It was a great success, recognized by Bill Gates. Yeah, because sometimes Bill Crosby, but it's a little bit different. Bill Gates, who gave a lot of money to this kind of experiments, and they've been done in Vietnam, Indonesia, Brazil, Colombia, and now in the United States, just to eradicate the dengue. And there is a consortium called, it's here in eliminatedengue.com. And what they have, they infected the mosquitoes which are in yellow, and they release them in nature. In Montreal, they used to fly, but here they do not fly. So, sorry. So the summary for the third objective, we are almost at the end. Wolbachia and arthropods, it's a master manipulator. It's called, it has a lot of names. I didn't tell you all the names. A master manipulator of reproductive processes in arthropod, this ensures its own survival. Experimental infection with Wolbachia of non-natural infected insects or with new strains of natural infected insects affect the ability of insects to transmit important pathogens. Several strategies have been developed and tested in field conditions to suppress mosquitoes and other medically important insects. In conclusion, Wolbachia has an important role in the pathogenesis of filariasis and represents a target for chemotherapy. The antibiotic treatment should become universal. Its presence in insect, either natural or experimental infected, represents potential means of controlling insect-borne diseases. It has a great potential to limit and even eliminate several devastating human illnesses. And I thank you very much if we have questions. By the way, this is an insect egg infected with Wolbachia. Thank you. Yes.
Thank you very much for a very informative lecture. I was so impressed by your lecture. So I have a couple of questions. So you mentioned that the Wolbachia cannot be cultured by in a pure you can't cultivate.
To culture it?
Yeah.
Yes, you may culture, and it was even proven that you may culture it on some human cells.
So, but
cells lines.
it cannot grow by themselves.
No, but if you add everything which it needs, could be cultured. But in all the experiments here is just PCR.
Just PCR. So
first identification with electron microscopy, then immunohistochemistry with the antibody against the Wolbachia surface protein, but PCR is the method used to study.
So my question is about evolution. So so many insect species are infected with Wolbachia, but Wolbachia cannot survive by themselves in the natural conditions.
This is why it uses the reproductive tract to multiplicate and to disseminate.
Yeah, but reproduction system cannot beyond the hurdle of species.
No, it cannot exist without a host. Should be somewhere, either in filaria, either in insects. Mm-hmm. Otherwise, they cannot exist in nature.
No, yes.
However, there is the...
my question is, how do you transmit over the species?
99%, this is horizontal.
Yeah, horizontal. How?
Yeah, how? One insect dies, mm-hmm. and the other one which comes there on the dead insect could have Wolbachia. This was proven for freshwater crustaceous isopod terrestre, which could have a wound and let his hemolymph, and the other will come and will be infected with Wolbachia. It was demonstrated by a French group in this isopod terrestre, also from the fresh water.
Okay, the next question is on the WSP, surface protein.
What?
Wolbachia surface protein, WSP. You mentioned the protein, surface protein regulates host immunity.
Yes, it's immunogenic and pathogenic.
Yeah.
And what we see in humans with the filaria infection is due to Wolbachia. It's not due to the filarial orb. Filarial orb is covered by the proteins of Wolbachia.
But Wolbachia is living inside cells. So how this protein is secreted outside the
it's different than mitochondria. Mitochondria import proteins while Wolbachia export the proteins. And they are not just intracellular, they are in the somatic tissue between the cells, and they go to the surface of the core of the filarial nematode. They are very intelligent.
I'm sorry, I couldn't follow. The protein was produced by Wolbachia and it is transmitted to the mitochondria?
No. I said they are like mitochondria.
Like mitochondria.
But the difference is between mitochondria and Wolbachia, which will be the future mitochondria, is that mitochondria import a lot of substances, while Wolbachia export all those protein catalase,
but it is still inside cells.
Inside cells, but this goes also out of the cell.
How?
I don't know. I'm not a specialist of Wolbachia. I was just fascinated by this.
Major pathway for the secretion is vesicle transport. So that cytosolic protein doesn't go onto the vesicle transport system.
I think that it has also some proteins which will have like permeation pores in the cells, and we let those proteins go out. Because it's in the somatic cells, in the somatic tissues everywhere, not only in the cells. I thought you'd ask me about the infection of humans.
Okay. Thank you.
If no one is interested, I won't insist.
It is important for you to tell us how humans are infected.
So far, they are not infected. And I'll show you. In 1999, someone found DNA from Wolbachia, from Brugia malei, in plasma of infected individuals. It was an isolated report. Then I told you that could be with a lot of difficulties, culture on mammalian and human cell lines. Then from China comes a report at the beginning of this year showing that they found 16 years RNA, I put it in French here, of Wolbachia in the blood of febrile patients with leukemia. It was very well demonstrated. Now in 2001, it was a CDC investigative report on the web, and they said that the CDC sent a team in West Africa because there were only females in those villages, and all of them were alike, like cloned, like the feminization and parthenogenesis. But it was false. It was like Orson Welles did a few years ago about the extraterrestrial invasion. However, there is a book here, and I recommend it to you to read it. I read it. It's the Destiny of Fools. Because, how should I say, because the growth of the populations, they said we have to do something, and they managed to infect a few people, and then all the people were infected. The population of the globe here, you'll see in 2050 will be just 500 millions, and it was a great sterility pandemic. It's a fantastic. But you have all the explanation of the reproductive phenotypes there, and very well. How should I say? I read it in 1 weekend. Yeah. So that's it. Any other questions?
I'm sorry, perhaps I missed something. I'm not a specialist, but we understand. Perhaps you explain that the process of feminization, the molecular process, how these processes of feminization is induced by this bacteria.
Because the uninfected Wolbachia in those species, it's normal. They give normal, how should I say, males and females. Okay. However, when one is infected, it annihilates the androgenic system of the male. So everything will be feminized. And there is, the genes are already, how should I say, sequenced, the sex genes. And there are some males, I told you, which have feminization resistant genes. So to to counterbalance the effect of feminization. And they are very rare. I didn't show you in butterflies. It was described that diminished population of males from 50% to 5%. And this persisted for 5 years and people didn't understand why. And then finally they recuperate. In other insects, they showed that when they dosed this feminization, almost no males, they treated with doxycycline or sulfamethoxazole, I don't know why, and equilibrium was established. I have a lot of, I don't know if it's here. This and how to, no, this all right. Any other problems?